Hello and welcome to another episode of the Evolutionary Hologenomics Podcast. My name is Anna Verissimo. I'm your host. And today we have a very special episode because the podcast just turned 1. Time flies when you're having fun, I guess. So we released our very first episode in June 2021, and now it's June 2022. And we, for our anniversary, we have a very special guest with us today, Professor Tom Gilbert, who is the director of the Center for Evolutionary Hologenomics, without which we would not be here having this conversation today. So, Tom, thank you so much for joining us. Hi.
Hi, thank you for having me.
So it sounds so easy now that you have a center. It's like you snap your fingers and you get an excellence center, right? That's how it went. So can you tell us a little bit of, first of all, what was with the process and the idea, the ideation? How did you come up with this idea for the center? And then we can go a little bit into also what the process was like, but just the idea. How did you come up with this?
Yeah, I mean, it's a great question. And of course, it wasn't just snapping fingers, but really the center, which officially started in January 2020, was actually, I guess, the accumulation of about 5 or 6 years of thinking and ideas that kind of slowly dawned on not just me, but actually myself and some of the key members of the center, right? So if we go back in time about I guess like 2012, 13, 14, I was doing what I've always done, which is I was doing sort of comparative genomics of animals and occasionally plants, and all focus on the big animal, right? And I hadn't really thought about microbes, and there's a very simple reason for that. That is because when I was an undergraduate, on Friday afternoons, all Friday, all day for 3 years, I had to do microbiology, and I hated it. There's nothing worse than standing in a microbiology lab for 8 hours, playing around with things you can't really see and looking out the windows at the people outside in the parks having fun and stuff. So I was never interested in the microbes, right? And so I was very focused on the big things, the animals, the plants, and so on. But then actually, what actually happened was in about 2012, I was doing a lot of work on birds, and a collaborator of mine in the US came up to me and said, like, I know, Tom, you work on the animals, but I've got an interesting project for you. Will you consider? And he brought this famous vulture project to my mind. And basically, Gary, my collaborator in the US, was in the situation where he was allowed to sample some vultures that were being euthanized in various US cities. And he said, look, I mean, Tom, I know you do comparative genomics, and I know you've been looking at, what is the genetic basis of how certain animals live in the way they do. For example, in the vulture case, what kind of genes in a vulture allow it to eat the nasty rotting carrion we basically ascribe it to. But Gary said, but, there might be more to it. Are you not interested in how they're also, what's the result of eating the carrion? Do they get covered in nasty microbes, right? How do they live with these microbes on them? And so Gary persuaded me to start a study in collaboration with some microbiologists that I did know, to look at the microbes on the faces and in the guts of vultures. And so I was willing to try. I mean, I thought it was a cool enough study that I didn't have to worry about the microbes being boring. And the outcome of the study actually was that, yes, of course, we found all sorts of interesting microbes on the faces and in the guts of them, linked to the fact that they were eating carrion. And it's not very surprising. If you take a mouthful of nasty, rotten, bacteria-laden carrion, it will end up on your face and in your gut. But the one thing that really I found interesting was, as part of the study, we got our hands on some of exactly the same vulture species that were raised in a zoo where they've never been fed carrion, and they descend from vultures that have never been fed carrion. And they had exactly the same microbes on them. And this to me was weird. Why would the zoo vulture have a gut full of microbes we associate with nasty rotting meat, right? And it started to dawn in my mind, well, if it's maintaining this association, there's probably a reason for that. Life doesn't normally keep things around that do nothing. And this is what basically finally got me, as an organismal centered biologist, to realize the actual answer to the question of how does a vulture eat nasty rotting carrion is, of course, there are genes in the vulture that allow it to deal with the nasty rotting stuff. But probably these microbes that it's deliberately maintaining are probably playing a role. And so together, the union of the two is the answer of how does the vulture fill the niche of eating rotting carrion. Now, I want to be very clear here. This is to some degree, the fact I saw this back in 2013 is really a reflection on how close-minded I was, because, people who work on cows, of course, have known for decades, hundreds of years that a cow eats grass because it's got the cow part, but also the microbes inside it, right? But it really took this striking example to really help me understand that many of the questions I was interested in before, like what is the genetic basis of how species bird X fills diet X, cannot be answered alone with just the bird. We need the two together. And conveniently at this time, technology was changing enough that we could start to actually think, well, maybe it's time now to propose that if we want to keep looking at these big questions in animal or plant or fungi evolution, whatever, we have to stop being so host focused. But similarly, the microbiologists have to stop being microbiologist focused. We've got to put two together. And this, of course, is what hologenomics is, right? It's just studying the complete gene content of an organism and all its little microbes together, and working out how they work together in order to answer whatever question you're looking at. But then, of course, it raises the question, what are the questions you're looking at? And this is actually, I think, the beauty of the fact we got our center. We are in the privileged position to be able to explore a very wide range of questions linked to hosts and their microbes, because that's what we do, right? We can, of course, study what I call the stamp collecting questions, where you pick one particular animal and it's one particular thing and find the answer for it. So which genes in the vulture and which microbes in the vulture allow it to eat carrion. But you can go big picture and say, what are the general patterns out there, right? Can we extrapolate from this and maybe start to ask some of the more deeper questions in ecology and evolution? For example, why are certain lineages of animals much more successful than other ones, right? Is it to do with the way their microbes and the host interact together? Or why is it some species are at risk of extinction and other ones aren't? Is it maybe the union of the host and the microbes? And to me, that's what's fascinating. How can we take this understanding, which has been around for a long time, that hosts do near their microbes, and use it to go back and address some of these much more basic questions that rarely get looked at, or in fact never get looked at in these contexts? And that was what the center was. The original grant application was myself and the co-applicants shown here, Anders and Rob and Alina and Sham and Ida and Anton and Morten. And I want to be very clear, of this lineup, basically Sham, Anton and Morten were the guys that, through discussion together, myself, and then it had dawned on us collectively that we could really propose this whole center idea, right? They were the guys that shaped the groundwork. But we also knew that to do this, we needed certain other people involved, and Anders and Rob and Alina and Ida bring various technical things that we needed to do this. So it was a union of these ideas and these technicalities that allow us to build the center. And so it started, and but the center started then, and fortunately it allowed several things. It allowed us to hire the first cohort of people, but it also actually gave us credibility. And this is key. When we proposed the center the first time, we didn't get it funded, partly because it was such a new idea that the reviewers were like, this is craziness. Why do you need to make life complex and go this way, right? And we had to then try again, and we had to think about how do we propose it to persuade the foundation. Sure, it's different, but it's worth funding. And one of the actually very big surprises to me was that the foundation chose to fund it. And I've actually talked to the foundation after, and I was like, well, just out of interest, why did you choose to fund this? Because the foundation asked me in the interview, how are you different to anybody else doing microbiome stuff? And my answer was, well, basically, we're proposing something everyone else thinks is crazy because it's different. And then I thought, that's it, we're not going to get funded. But to their great credit, the board of the foundation were like, well, sometimes it's worth going with these alternate ideas, right? Because if they're right, they open up things. And so that happened. It set the foundation for the initial work, but of course, it also set the foundation to expand. And we've been really, really fortunate that we've been able to both attract more funding to expand what we're doing, and we come to that later, but also expand the team. So actually, the PI team today is at least 5 people bigger, because we, once we started, we realized there are other very capable people around whose ideas fit with what we're doing. And so today we are, however many PIs are shown here, 2, 4, 6, 7, 8, 9, 13, including me, PIs, right? Which is great. It shows the applicability of the methods and stuff.
Yeah, so this is, we started with the milestone that is the podcast turning one, but there's also another milestone here, which is basically 10 years. You mentioned, you referred back all the way back to 2012 when you were starting to think not just the big animals, but actually the bacteria and the bigger picture. And then 10 years on, here we are. And within these 10 years, you said you applied the first time and it did not get funded. And even now, because we work on this field, we are still, it's more accepted and there's more people worldwide working on this, but still sometimes other people need convincing. So it's not super established yet. So what is the timeline? So what year did you apply the first time? And then what year did you apply when you got it? And then finally the center was able to start.
Yeah, so I think, so the application process itself is a two-step process, right? So you write like a preliminary one, and then a number of those go through to the second round to like a longer one. So it takes like at least a year from the very start to what goes on. And I think we basically applied the first time in maybe 2017, and maybe early 2018 we were told, okay, like, that's this time not going to happen. And then I think we applied again late 2018, and I think it was late 2019 that we had the good news that it was funded, right? And then it started very soon after in January 2020, spectacularly badly timed given COVID, because the first thing you do is start hiring people who then got hired to start in the middle of COVID or so on. But that was basically the process. And it was actually a very painless progress in many ways. Even when we applied the second time, we were tweaking what we did the first time. We looked at, the first time I think we were a bit too sure that people would accept our ideas. So actually most of the changes the second time were directly confronting what criticism we thought we would get as part of the writing, and maybe being a bit more diplomatic in the wording about why we thought our thing was worth doing. I've learned it's very bad generally to write and say wording which sounds like all this work has been done by others, they're wrong, we have an alternative. Of course, the correct way to write is an amazing foundation has been set by all these other people. We have a few tweaks we can put on top of it, right? So, but of course, I mean, everything we do is based on the foundation of other people, whether they were right or wrong, or whether their model was fully developed or not. A lot of work has got in, right? And one must always give credit for that. And I personally, I'm sure, have been maybe a little bit too not sensitive enough in maybe the way we've written stuff before, right? Which is, lesson you learn. But anyway, so yes, it was funded then, and the first grant was funded for 6 years. So we end in 2025, but you can apply for a 4-year extension, and that itself is an interesting process because the extension has to be thematically linked, but not, of course, doing what you've already done, right? So the expectation is you develop new ideas during the time. And actually, that's what's so nice about getting new people on board with new ideas, right? We're kind of laying the groundwork for logical extensions, right? And you mentioned, of course, that not everyone accepts the kind of way, but a lot of people are starting to do it, right? And not only do we, of course, talk about the ideas a lot, but other people have the same kind of realizations in parallel, right? And we've written opinion articles and serve others, and it builds together a community, right? And to actually jump to the last slide I was going to give, actually this year in September, we're actually holding what is really the first hologenomics conference, right? Which is really exciting that we can actually put on a conference and bring together a wide range of people to actually do this stuff in this way, right? And we are very inclusive. We believe basically we're not doing anything particularly exceptional. We're doing things in a certain way and setting a framework that we hope many other people in many other places can benefit from, right? Because we've reached an era where technology, both sequencing technology, but also just the basic lab technology, but also the computational technology, is improving to the extent where it is more and more and more feasible to generate the kind of data sets we do. That doesn't necessarily mean it's any easier to analyze the data set, right? But at least it's, we're not in the era where it was it's so prohibitively expensive you can't even consider that. Right now we can consider it. It's so people can at least embrace the option if they want to.
Yeah, and we've seen that a lot in science when a lot of things are now being revisited because the knowledge has increased, the technology has advanced. So there's a lot in drug discovery, for example, or testing old treatments or old drugs for other things and so on, or analyzing now the mechanisms of action. And so, as you mentioned, now it's making sure that all these people from various fields, because now everything in science is multidisciplinary, that all this knowledge comes together to create new combined knowledge. But so you mentioned that there were some comments and reviews to the first application that failed, and then you built on that to make it better. So could you just let us know what was kind of the main thing that people were against or didn't believe in the first application, and then what were the actual original aims that you set out for these first 6 years of the center?
Yeah, so the original application had three reviewers, and one of them loved it. That was great. One of them was like, yeah, I mean, it seems okay, but basically, aren't there a load of people doing microbiome stuff already? And they kind of didn't appreciate maybe maybe not appreciate, but maybe we hadn't sold the subtlety well enough. They just thought it's just another microbiome collecting thing, which we're not, right? The third reviewer, which I think, of course, was the most problematic for us, was actually to do with the name of the center. Our center is called the Center for Evolutionary Hologenomics. And there is there is a passionate debate between a small number of people in holobiont theory about whether the holobiont, and the holobiont is the host and the microbe together, whether the holobiont is under natural selection or not. This is among evolutionary theory people, there's a lot of enthusiastic discussion about this, and a lot of people think it's not, and they're backward and forward and so on. Right now, we're not working on, is the holobiont under evolutionary pressure, right? Is it under natural selection? That's not what we're working on. But of course, evolutionary hologenomics sounds like it. And unfortunately for us, one of the reviewers is one of the guys who hates the idea that a holobiont might be under natural selection. Basically, the review went along the lines of, it's a stupid idea. The holobiont's not under natural selection. This is a waste of time, which, again, can either be viewed as we didn't do a good enough job presenting what we wanted to do, or there was a reviewer with some kind of preconception about what we wanted to do, right? And kind of didn't actually look at what we wanted to do. So actually, the second time round when we wrote it, the major change we wrote was like, we are writing about evolutionary hologenomics, which is how does the union of the host and the microbe explain how things evolve. And then we said deliberately, just to be clear, this is not the same as is the holobiont under natural selection, which is a very interesting blah, blah, blah. But to be honest, the answer doesn't matter with regards to what we want to do. We actually tackled that head on, right? And that was mainly the main change, right? And it was like just clarifying a few things or so on. So the actual underlying aims of what we wanted to do didn't really change. And there was basically 5, what we called, I think, work packages or research lines that we said we wanted to do. And we said, look, this is a new kind of thing. And number one, we need to set a framework. We need to basically generate the lab methods and the computational tools to deal with this kind of data. And it's very methods, right? But without the methods, you can't build on stuff. And also linked to that is, let's put out a few opinion or theory or like ideas papers, right? And then the next 4 were much more, I guess, research based. And one of them was actually more about the theory and like the ideas and the models, like exactly how might this thing work and so on, and the kind of the math behind it and so on, and the general framework. One of them was about what is the actual relationship between the genome and the microbiome, and how does genetic variation at the host level shape the microbiome, and how might that set up what else can happen. One of them was actually about, can we look at the relationship between the microbiome and the host in certain situations of interest? For example, if you have a certain fish that's adapting in a certain environment and other fish not, what's exactly going on there? Can we find the mechanism? And the very last one was more bigger picture. Can we take that and extrapolate it to the context, right? And more general thing. Sorry, I actually forgot there was another one on top of that I'd forgotten about because it's a bit delayed now. The very last one was, can we then take it to to the human, but not in the medical context, in what we call the extended holobiont or hologenome. And what that is we typically view the hologenome as the host and the microbes inside it, right? But what humans and a number of other species have done is sort of move microbes outside the body. So fermentation, what Rob Dunn is very interested in, is us using microbes for our good, but outside of the body, right? You know, we take whatever, like some kind of plant, and we ferment it and we make it edible, right? So it's moving beyond microbes inside us doing it to kind of creating the environment to do that, right? So that's the very, very extension. And that is actually the last work package to be started. Today is the 2nd of June, and yesterday, the 1st of June, the first postdoc and PhD were just hired to work on that with Robert and with Sandra, right? So that's going to be exciting to see how that goes. But that allows us to kind of go from the natural system to the kind of extended one that humans and other species can do. So that's what started. And, you know, we have, of course, made some progress. One thing that's really a privilege when you get a center grant is you can start to buy things that you need to do things on the big scale, right? So I'm just showing here 2 examples of shiny things. But, you know, you can start buying robots to help upscale things. That's not the same as getting the robots to work, as many people know, but at least you can have them. But I mean, you know, this machine on the left here, the Cyprint, this is the kind of thing I never knew even existed till Anton came along. You know, it's a machine that's only function is to open tubes, fill them with liquids, close tubes, and label them, right? I mean, such things exist, but of course, once you want to do stuff on the big scale, you can waste huge amounts of time manually doing that yourself, or you can buy a nice machine to do that. So, you know, the infrastructure, the computers, the machines that allow us to do more stuff, you know, they were provided by it. And as I said, we can definitely, you know, work on models and ideas. So all this is showing here is that how our ideas are evolving about what exactly is the microbiome and how do we measure the microbiome and how does it change through time and how can you model that? I'm not going to go into the details, but you know, finally, because we have money to hire people to think about this stuff, you can dedicate your time to doing this, as opposed to what happened before, when it's a niche thing you're thinking about on the back of other grants you're doing, right? So the privilege to actually spend time thinking about this and writing ideas and so on. Of course, we could start doing some of the experiments. I mean, what this shows is Louisa, one of our PhD students, down at the Zebra lab, which Anton and Astaiska created down in the Basque Country. You know, Louisa, several people have been down there, and you can start collecting, or even actually in this case, buying lab mice and doing all sorts of experiments where you change the environment and you see how the microbes change and you can see how that affects the behavior and start tackling the question of how are they adapting? How is their adaptive potential going? And of course, we're doing this on many, many systems now, thanks to the framework we have to then take the data and do stuff with it. And actually, another thing that's really nice to see, the long term of the center, the fact that we have at least 6 years and hopefully 10 years, and we have a degree of credibility that means we can apply for other grants, means that bigger initiatives can start. So what I'm showing here is Anton's baby, the Earth Hologenome Initiative. And, you know, Anton has basically set up this huge global collaboration coordinated by his team, where they're tracking down researchers all across the world who are going to the field anyway to do whatever they do in the field. And as long as it involves some degree of trapping animals, Anton's team send them these amazing kits where they can take gut samples or fecal samples or tissue samples, depending on what they're doing, in a very, very standardized manner, send them back to Copenhagen so we can generate microbiomes in a completely standardized way that can then be used for many of these bigger picture questions, right? And this may sound like a nerdy thing, but it's not. The biggest problem in microbiome work is actually standardization. As those who work with microbiomes know, if you do anything different from how you collect it to what you store it into to how you treat it in the lab, you get all sorts of noise that makes it really hard to compare the results. And the ability to have a standardized thing like this is just going to be an incredible, incredible treasure for the future. But that requires long-term funding and infrastructure. That sexy tube labeling machine is exactly for this kind of thing. So you've got to label thousands and thousands of tubes, right? So you know, without the framework, you can't think long-term, right? And that opens up other things. And another thing I want to flag, and this is very much, again, the baby of a number of PIs, Morten and Sham and Anton and Astaiska and Miyako, who was here, and I'm sure others, we can actually try and spread the news. So we did run last year, 2021, a PhD course on hologenomics, right? It was run at KU for the first time, and to try to spread the knowledge. And again, it's the kind of thing you can start doing once there's enough people around to actually talk about interesting stuff, right? And so that to me is a really, really great thing to be able to see happening, these kind of educational option. And of course, we can also, you know, we get visitors coming the whole time, we get people applying for grants to work with us and that kind of thing. And that leads to, on the positive side, lots of exciting new stuff. On the negative side, desk headaches from Morton, the head of section, trying to find a place to put them all. But it's also nice to be able to run these kind of, courses that people can come in and learn from and go away, and hopefully spread the idea of what's going on.
Yeah, so a lot of different things. So obviously there was the initial people, and then lots of new people joined that add a slightly different angle, which is great. And actually, I'm just going to do some shameless promotion now, because you mentioned the Earth Hologenome Initiative, and that was actually our very first podcast episode. So for the people who are watching or listening, if you haven't watched or listened to the other podcast episodes, so the very first one was on the Earth Hologenome Initiative. So it was a very nice chat with Aoife, who manages the project. Then the second episode was on the Horizon 2020 project, 3D Omics. So I guess that's, you briefly mentioned, that's one of the great things that the center also allowed to do, because you had the center, you had all these people that were going to work together and collaborate, and then they could expand the international network. So we actually got a few big projects funded on top of that. One of them was 3D Omics, which takes all the layers, all the omics layers, and then will try to also add the three-dimensional aspect to it. So what bacteria are there? Where are they exactly? What are they doing? How does that affect their environment, the gut environment? And then how does that affect the host? So if you haven't watched it, go watch that one. Then we had three more, one on marsupials and patch microbiome, which was super interesting with Raphael. Then Jayella talked to us about salmon and the tapeworm microbiome. So there was a very nice analogy about the matryoshka type thing, where you have an animal inside an animal, and then each one has their own microbiome. And finally, we switched it up a little bit in the last episode, and we had a journal club with Jacob, one of our PhD students. He's about to finish his PhD, I think, on food additives, gut microbiome, and trout health and disease. So that was super interesting. We kind of went through two of his papers that just got out. So there's so much going on at the center, because we have so many people and we have all these different fields. And one of the things I always find fascinating is when you do these kind of elevator pitch promo talks, and you show that graph of how many publications we had for this center. And basically there was not much, and we weren't really experts or, when the center started, but then there is this kind of almost exponential curve of achievement. Can you tell us a little bit about that? Maybe some highlights?
Sure. Sorry, door just rang. Absolutely. I mean, it's. I want to mention one thing actually first that I forgot to mention, but I do think it's important. You mentioned we got all these spin-off grants, right? And these big EU grants, and they are very much. The credit goes, of course, to the co-PIs who did it. But one of the nice things about the center, at least 3 or 4 of the co-PIs have their job because of the center. Right? Had the Grundforskning Fund who funded us not believed in the idea and provided the money, I wouldn't have been able to lobby to the institute to create these faculty positions to do this whole thing, right? So, and that of course then allows these big grants to come in and so on. And that comes to your final point here, because when we started, we had very few papers because, it was ideas we had. We'd never worked on this. We were essentially jumping to a whole new field, right? We didn't have we weren't like a super tanker already going on, pumping stuff out. We had to start from scratch. But of course, the enthusiasm of these people who basically dedicate all their time to the center because it's their baby means that, of course, they start pumping things out. And the papers have been increasing quickly. And the grants as well. I mean, this was a very nice large grant, but the number of grants we've got since, the co-PIs based upon this framework is going on. And that's what's really great to see, this increasing amount of stuff and the spin-offs to the new areas and so on, right? I want to say one thing though. I mean, we weren't experts at the start, and to some degree, the one, I guess, frustration, if you want to call it that, although it's not the way science works, is that one thing we've learned is how little we know, right? I mean, it's one thing to say we're going to jump ship from this animal comparative genomics, evolutionary genomics we do to do microbiome stuff. But of course, we're jumping into an area that's very, very, very complex, right? And we are learning all the time how little we know. And that's part of the reason we have to get new grants to try and solve these things. So Finding Pheno, for example, one of the spin-off projects, is a computational grant born because basically we realized we don't really know, nobody knows computationally how to deal with these kind of data sets, right? 3D-omics was born because basically we realized, well, look, we're treating the animal gut as a black box, and we just get the microbes out, and is it really a black box, right? Do we need to make things more and more subtle, right? Because of course, the more accurate a model is, and so on. So we've encountered a lot of challenges that have to be solved. And the exciting thing, of course, is that we're in a position to try and solve them, but also we're in the position to tell other people everywhere about them in the hope they can solve them. And some of the challenges are very mundane. I mean, simply, can we get enough computing power? Or more importantly, can we afford to pay for the computing power to deal with the data, right? I mean, the bills we sometimes get to pay for the supercomputing and the storage are just massive, right? And some are mundane, like can we get the bloody robots to finally work and do the kind of stuff? But more actually really fundamental to what we're doing. So what this figure shows here, especially the top A, B, C, and D, is different ways to view a microbiome. We generate microbes from a host. We get a lot of sequence, but how do we represent that information? Do we, as in A here, just represent it as, what A shows is the microbes that are inside the individual, but what B shows is, we could represent it as what microbes are there and what the abundance is of the microbes, right? There's more green than blue, whatever. But, C and D represent other kind of representations. C, for example, might represent what genes are present within the microbes, not what the microbes are, but, what genes are in the system. D might represent the activity of the genes, so what genes are being used. And until you can decide how to represent them, you can't even model how things change, which is what the second row represents, right? And this is fundamentally important because this is the basic thing we're measuring. And if we, and not just us, if the broader microbiome community is not yet sure what is the best way to represent it, and that itself may change by scenario, of course, everything else you're trying to do with this gets very, very hard, right? So I think what we did, and we were naive, we were new to this because we hadn't done it. One of the biggest problems was we had really not thought enough or realized how hard it is to generate this baseline information. It's not just as in an animal sequence the genome and you got its genome, right? And so that I think is going to be a major thing for us to think about for a long time. And of course, that requires all sorts of grants and labor. And of course, a lot of these things became apparent after we started projects generating the data. So the people we hired originally are working on the actual projects, right? So it opens up other kind of things. But this is also the excitement, right, about this. There is a lot to be done in this very nerdy details thing. And that doesn't just benefit us, and and it's not just done by us, but whoever does the work will benefit a lot of people, and being able to watch how that changes, and having people who are much more skilled at Denai who actually understand this and who are working on that is one of the real privileges to see, right? See how this goes forward.
Yeah. So, and it's good that you mentioned computational power, because we actually, thinking about the previous episodes of the podcast, we pretty much everyone who was on the podcast is a bioinformatician. Some of them are purely bioinformaticians, some of them do a bit of lab work and then also the data analysis, and they make it seem very easy. They just say, oh, we got we did this with the samples and then we just sequenced it, analyzed the data, and here's the result. But what happens between getting immense amounts of data that are very, very complex data sets, between that and actually making something out of them and having results in the end and having answers to scientific questions is a lot of work. And obviously, as you said, technology has evolved and there has been a lot of software developed and a lot of code developed, and there's a whole community around that. But still to this day, there's a lot of things that we're only starting to kind of figure out how to analyze, and some things we don't know how to analyze. So it's very nice to see that at the center we have a lot of people in the lab generating the data, and it all starts with scientific questions, of course. But then we have a whole team of dry lab people in the computer, and we have very, very good bioinformaticians with us who are actually not only applying the known methods, but also coming up with the new methods to actually analyze the data that we have, and finding pheno, I guess it's one of those examples in Sham's group.
Yeah, I mean, you're right. And, one of, again, the nice things about having a center which is of a sufficient size is you can have both parts of the equation. Right? And that's super important, because I'm a lab person, right? I have enormous respect for any lab person who dares to jump into computing. Right? I never did it myself when I was younger. But I also have enormous respect for computing people who actually take the steps to understand the lab challenges, right? Because without understanding the full thing, you end the risk of following pipelines, and pipelines have got their time and place, but what pipelines don't teach you is the challenges, and without challenges, you don't know the risks and what can go wrong, and you're at risk of generating results which may be flawed due to some set of the process, right? So actually, the ideal situation is that the researchers from one or the other end of the spectrum kind of get the full information, right? But of course, then you can't specialize. And of course, it's a very challenging thing. And that's why I say I have enormous respect for anybody who is willing to make themselves uncomfortable and try and do that, right? Because in the long run, though, I think they learn a lot more, and they might never become the full-on specialist in it, but knowing enough to know where the problems are is really, really key, right? So again, that's one of the very nice things that we got the Center funded. We can have these people together, right? And I always love it when I see what I love is that when one of the two parties appreciates that it's not easy for the other party, right? And realize, they're just not the only ones doing the hard thing, and they talk together and they discuss the challenges and so on. But it's certainly not easy. And I say this, I am not that old, but I'm old enough that when I was doing my PhD and postdoc, this was pre-next gen sequencing, so I never needed computing, right? I mean, I opened things on a computer, looked at pictures, whatever, right? Looked at sequences manually. So I never had the need to jump into computation. And by the time the computation came along, I had too much other stuff to do that I didn't have the time to learn it. So that, it's really great to see these people here, and I have enormous respect for what they're doing.
Yeah, and as you know, I love communications, and I think this kind of also gives rise to another type of communication, because people think science communication is all about making very complex things simple for the general public. But actually, nowadays, in these very multidisciplinary teams, it's also important to use that kind of science communication in a way that you make your subject simple, so that your colleagues actually can understand what you're doing, so that you both can understand what each other is doing and then work together in the same project. So that's very important, that people try to make it simple to make it work.
No, I 100% agree. And that's another thing I would like to say that's really nice about what the Center allowed, because as you know, we have a number of people that basically specialize in communication, right? Either because they've been hired on some of the EU projects, or because they were part of the original Center, right? And that allows us, of course, to have just the regular newsletters and stuff, which are super, super nice because they keep everybody connected. But they also exist to help or encourage the staff members, the scientists, to actually participate in communication and learn how to communicate at the right level, which is by no means easy, right? Putting yourself in the mindset of an audience who don't have the background is a real specialized skill, right? But once you have it, it opens up so much potential, right? So I'm always extremely happy when I hear that anybody, especially one of the scientists, has taken the time to volunteer in something, whether it's walking around Sonnemarken at the Bloom Festival with students, or doing a podcast, or doing a high school thing, right? But that can only happen when the other people in the center, people like you and Christina and Thomas, who just left us, and Anna, for example, help make those connections, right? And encourage them. So it's really great to see that. And again, that comes because of the resource we have, that is the center. So it's, when I applied again for the center, and when I was having the negotiations, I think the director of the Grundforskning Fund was going through the budget and said, well, I can see you've written in like a full-time communication person. Why is that important, right? And I said, well, because that will make the communication happy, and I'm sure you'll be happy. And they were happy with it. And one of the really good feedback I got after the first annual meeting where the foundation came, we talked about all the kind of stuff we're doing, and they were really happy to see that, right? They were happy to see the engagement. I think I mentioned this just the other day in a section meeting, but we do have a monthly newsletter that of course takes a lot of people, a lot of work. And of course, one has to hope it's actually read and appreciated, right? And apparently, we can track this, and it's good to see that a good 70% of the recipients actually are opening it and hopefully enjoying it, because it's always nice to know things you are doing or that other people are doing are being valued, right? Otherwise, why spend your time on that? So, yeah, it's all very good, and it's all very important because there is the future, the expansion. How do we go forward? And one can only expand if people believe in what you're doing, if people have heard about it. And a lot of the expansion we've done, either bringing in new PIs or exploring new areas, is because people have either heard what we're doing. I'll give you a very good example, actually, in this regard. I mean, Sandra, who joined us as we were starting, Sandra had applied for grants in biology to actually be doing work on humans and Helicobacter in the gut and mice in Helicobacter. And she actually then heard what we were doing and came for a chat, right? And then she realized, well, I mean, what she was doing would be very complementary with what we were doing, right? So luckily we were able to bring her in, and in doing so, expand from the more basic stuff to the more applied health relevant stuff, right? And of course, that opens a lot of potential, but that would never have happened without this initial kind of stuff. And every time Sandra goes out there and talks at Venum Videnscape or whatever she does and spreads it on, that brings in more connections, right? And another thing that we've been doing again is we've been successful in getting spin-off grants, and this is just 3 examples of Steisker and Sandra and Morton, to try and bring in new technology. There is all sorts of crazy biotech technology out there. I mean, there's organoids making little tiny organs in 2D, or even gut on a chip, which is what's on the right here, right? And CRISPR, what Morton's guys are working with. And, because of the outreach we're doing, we make these connections, and suddenly through these connections, you can start benefiting from all this expertise elsewhere, right? And bringing these things in that opens up the potential from what we can do. And it really takes the work to a whole new level, right? So, but that all comes from the connection. And just another example, thanks to the initial outreach, we got talking with the Carlsberg Research Labs, right? That led to various projects that myself and Chris here, Chris is one of the other new PIs, started doing with plants. I mean, our center's all animals to start with, right? But there's no reason why you can't do hologenomics on plants, right? And so, we've had some recent projects on maize, but now Chris has got really fun projects on hops with the Carlsberg Labs, right? So it's really nice to see these. And one last example, because I do want to mention this, Thomas and Bent were already here at the Globe, but they make tools to work on phages, and we kind of ignored the phages, right? That's just another complicated layer, right? But it became very clear that if we want to understand what the microbiome is doing and what regulates it, we can't ignore that angle there, right? So it was a very natural extension to bring the phages in. So we're really covering the full community of what's going on. So that's the exciting thing. The outreach brings more potential, and then you can really do all sorts of fun new things.
Yeah, definitely. And just one thing that occurred to me when we were talking about, how the landscape has changed from one PI writing all the grants and trying to be a project manager and trying to be a supervisor and teaching and doing all these million things. They still work very hard, but definitely with these bigger projects now, one thing that changed and is still changing, and people are still trying to figure out how to make it all work with the rest of the team, is that nowadays these big projects also come with a project manager and an outreach manager. Sometimes it's the same person, sometimes it's different people, but this is something that didn't exist before. And because the projects are so big, so these Horizon 2020 and soon Horizon Europe, they have 12 or more different partners all over the world. They have huge budgets. They have very ambitious aims, but they also come with legal issues. So now universities have EU offices to help with legal and finance and EU rules and all these things. So there's this whole
very demanding
village.
EU project officers.
Sorry?
Very strict EU project officers that you have
yes, indeed. But then we have a whole team of liaison officers and so on to help us navigate. But it's just how things evolve, right? As big as it gets bigger and more complex, then we need all these people with extra knowledge that it's not just science, but it's actual legal, because then it's about contracts and MTAs and intellectual property that can be spin-offs, new products, who knows. So it's an exciting new era where all this now is possible with so many people and skills, and the center is doing well. It's not even halfway through the first 6 years, right?
Yeah, we're like 2.5 in basically. But I mean, yeah, and one has to be clear. It's not because a lot of that started from day one. You know, we were trying for many things at once, but the success came from them being merged in one framework, right? But of course, you're right. I mean, it's really important to realize that things like this can only exist with the support of everyone, from like, Cecilia, who's running the administration, to the project officers, like you said. But there's also the research assistants and the lab managers. I mean, because there's all these little bits you never think about when you're a smaller unit that have to be in place there. And then that brings other challenges, because of course, when you have many people, they can either work harmoniously or disharmoniously. And so one has to make a situation where the communication is so important, right, to make sure things work together. But I think generally, when people are unified in a vision, and when people realize that we're all working towards the same goal, there's just a lot of potential there, right? And whether that's within the center or within the institute or within the networks of all these partners, right? To me, that's so important, that there is this very good atmosphere where people benefit from the experience of each other, right, and build up synergy. And I've never been a believer in competition. I've always believed that when scientists compete, it's typically because they didn't know what each other were doing, and it's very rarely deliberate, right? But I know other scientists disagree and think there's a lot of deliberate. But I'm a believer that science is about collaboration, and typically, if I ever see anything that looks like it's competition, it's rarely deliberate competition. And often, the moment you talk to people, you get even more synergy, right? And so that again is why communication is so important, right? So people know what you're doing. And I'm actually, to be honest, always relieved when somebody comes to me with an idea and they want to do stuff with us. It's like, great, we have so much data, please take data, take samples, do more with it, right? That's much better than, oh my God, they're stepping on our terrain, right? There's a lot of stuff that can be done out there, and the more people that work together, the better. So it is always a great pleasure for me to see when things are working nicely, and that luckily is the way things are. And let's hope they stay that way.
Definitely. And you just mentioned yet another new thing that wasn't a thing, or still isn't a thing, unfortunately, in some places for various reasons, which is the culture and the community, the spirit of community within your workplace and mental health and all these things. So even though the center started with everyone kind of from home and didn't really had its grand inauguration because everyone was home, we did have a retreat, and I think everyone really appreciated that there was a bit of science, but it was mostly about the people, and everyone was just so happy, and I think everyone felt like a center. And there was some facilitation, so there was an external speaker on how to understand other cultures and so on, because the center is so multicultural. So do you know how many nationalities we have at the moment?
I have no idea, but it's a lot.
It's a lot.
But there are very few of the same nationality, put it that way. So, we have a very high, beta diversity, very high beta diversity or something. Anyway, but there's a lot of people. No, but you're right. It's very important to do this, and I'm very glad we could have the retreat away, and I learned a lot from it. I was surprised at certain things. I never in a million years thought Anton would get people to do folk dancing of different cultures, but I was a little bit disturbed at the level of cheating in the hologenomics game, and I thought my center members were much more followers of the rules, but anarchy can be good.
It's called strategy, Tom.
Strategy. I learned lessons about who likes to party sometimes and have lots to drink, and it was very fun. It was glad it worked, and let's hope we can do that again, right? It's hopefully there will be other events. I mean, a lot of us are going to this conference in the Basque Country soon, which should be quite a milestone event, right? Because it's getting together not just us, but people on the outside, and we'll forge new connections. And at some point, we might actually hold like a smaller hologenomics conference in Denmark. We were meant to do it originally, and then COVID made that impossible. But at some point, that would go on. So there should be lots of other things, and I'm very excited about all the future. Oh, I just want to show this slide, because for those that don't know, this is the official advert for our conference, which I encourage people to look at going to. It's still possible to get in.
Yeah, and so for those who are listening and not watching, you can just Google Applied Hologenomics Conference, or just write Applied Hologenomics Conference altogether.eu, and you can find the information. It's in Bilbao, in the Basque Country, from the 13th to the 15th of September. So just to end, this was a really nice kind of full circle journey, and we're just beginning, because we're not even halfway through the first period of funding, which is 6 years. So Tom, would you like to just end with telling us either what is sort of the big next challenge or the big next steps after all this that people have now initiated and are working on, and we're kind of expanding all the time in terms of people as well as in terms of fields, until finally maybe we can take over the world? What is the next step and challenge?
Yeah, that's a tough question, because I mean, there are many challenges, but I actually think a real challenge, and this is very much due to flaws both I have, but also a number of my co-PIs who think like me have, is we get so excited by new technologies, right? It's so easy to apply for new things that are we fully leveraging on the data we generate already, right? Are we making sure we take all this data and do what we can with it? Because it's very easy to always jump to the next thing. I mean, and then, you want to do that because you can do things better. But of course, you've got to do that without losing sight of what you have already, right? So, and it's not about, I think, meeting the goals. It's about fully exploiting all these big data sets that have been generated, that have been collected, or so on, right? So balancing out those things, making progress, but without leaving what you have behind, without accumulating unfinished data sets, right? So, and I bring this up because it's a problem I've seen with my own. I mean, I'm sitting on some very big data sets from past projects that people left, or people moved on, or whatever, or we changed track halfway through, and I guess you could argue that can be quite wasteful, right? So hopefully we will be able to balance this, right, and continually exploit what we have and use what we have while also developing going forward. And I think I don't want to call it a challenge, because I don't think it will become a challenge, but I think it's a thing for us to not lose sight of, right? I mean, there's always new exciting stuff out there, and of course we should always do it, but one doesn't want to do it at the risk of missing out on really cool stuff that already exists, right? So balancing the two.
Yeah, and one of actually the great things about that, we did take some action. There was a workshop, a brainstorm session, how to align everyone's goals with the goals of the center, and there was a little bit of auction, offer and demand of the data sets and samples and so on. So it was really great to see all the different groups forming these new possible collaborations, where people have data sets and people who do dry lab work might want to analyze them either newly or actually to compare with other things and so on. So that was great to see. And another thing I just thought is, sometimes you get more than what you bargain for. So I remember the big Horizon 2020 project we had, and it's still ongoing, HoloFood. Even failed experiments can provide, so, failed because they were not the aim, but then they're an experiment in itself. And one good example is what we lovingly call the crappy fish. That was a failed experiment. And then I think so far, was it 3-4 projects that came out of it?
Crappy fish has been very, very good to us. So I think Anton deserves credit for the name, from what I remember. I thought it was me, but I'm just very good at stealing everyone else's glory. But Anton came that. But it's the realize. I mean, we could have just thrown it away, but luckily, it happened at the right time, and we had a great visiting Erasmus student who was willing to try it. And as you say, it's given a lot, right? And that's the point, that you need to think. I mean, often what people forget is the effort that goes into running experiments, getting them. I mean, these fish, they're a trial, a lot of money gone into time, the labor that's gone into collecting these kind of things. I mean, some of our chicken and salmon data sets from the applied projects, the number of man hours that have gone into just the sampling, right? They provide these incredible resources, and they're there to be used, right? And not just by us. I mean, I always encourage other people to consider, if you guys can use these, because they're there to be benefited. So yes. Yep. Before I go, I'd like to say one more thing. I would like to say thanks to you, Anna, for doing this. Your enthusiasm for these events, and I know not everyone knows this, but I encourage people to check out the Nerd Nights that Anna runs as well. There are plenty of things Anna's doing, and it's really great, Anna, that you have the enthusiasm for putting this together. So very kind of you.
Thank you so much, Tom. It was a great pleasure to have you in this 1 year anniversary. So we didn't have you for the first podcast episode. We have you now for the 1 year anniversary, and hopefully there will be many more to come with the success and the growth of the center. So thank you so much, and best of luck for the future endeavors, which are many, many.
Well, thanks to you.
Thank you.